Good morning, everyone, and thank you for joining us this morning for Winship Grand Rounds. A few housekeeping issues first. If you're an Emory University or healthcare employee and would like to receive a CME credit for your attendance, the login information can be found in the chat feature on the bottom of your screen. If you have any issues accessing that or getting CME, please send Julie Hawkins an email or drop a note in the chat feature. This morning, we're excited to welcome Dr. Alan Ho from Memorial Sloan Kettering Cancer Center. Dr. Ho has been the Geoffrey Beene Junior Faculty Chair and medical oncologist and is a medical oncologist at Memorial Sloan Kettering Cancer Center. He specializes in head and neck malignancies, and focuses on difficult malignancies, the rare tumors that we all have nightmares when we see and try to manage these patients. In addition, he focuses also on thyroid cancer. His research interests lie mostly in translating emerging scientific knowledge to these diseases into new drug therapies and new clinical approaches for treating these patients. Dr. Ho attended medical school at Washington University. He completed his residency at New York Presbyterian Weill Cornell Medical Center, and subsequently a fellowship at Memorial Sloan Kettering Cancer Center, where he stayed on faculty. I know that Alan has been very active in cooperative groups, including SWOG, including NRG, and has been a long-term friend and colleague, and we all look forward to his talk today. Welcome, Dr. Ho.
Thank you so much, Dr. Saba. Thank you so much for this invitation to be with you. Of course, I would like to be there with you in person, but nonetheless thrilled to have this opportunity to kind of speak and share some of the research that we've been doing and what's been going on in the field. And so, as mentioned, I'm a head and neck medical oncologist, but I do very much academically focus on therapeutic development for rare diseases of the head and neck. And that's what I'll be discussing today. Here are my disclosures, and we will be discussing off-label use of drugs. And so just to put into perspective, when we kind of think about the different primary malignancies that we treat on the service, of course, the most common are head and neck squamous cell carcinomas at just a little over 11 cases per 100,000. Thyroid cancers also, as mentioned, are not rare, but they're rare to the medical oncologist. So the REI refractory type thyroid cancers that we deal as medical oncologists and we try to develop therapeutics for are much less common, and of course results in a fifth of less deaths than you see in head and neck squamous cell carcinomas. And then salivary gland cancers, less than 5% of all head and neck malignancies, and of course occurring at 1 per 100,000. So academically, the clinical trials that I focus on and develop are really in RI refractory thyroid cancers and salivary gland cancers, but I'll also be sharing with you a story in head and neck squamous cell carcinomas where we're looking at a specific genomic subset of disease and therapies directed at those patients. So, I started on the service here a little more than a decade ago, and thinking about this focus in rare cancers. And many of the challenges in developing clinical trials in this space are really the typical ones that exist for all diseases in terms of thinking about feasibility and having the right number of patients for the right questions, single versus multi-institutional trials. You also have to deal with the fact that not a lot of clinical trial data exists for these diseases, as you know, the lack of historical data as comparators. And of course, in the beginning, availability of funding and interest, both from industry, particularly from industry, and educating people on the need and the importance of these questions, and then just the limit in biologic understanding. But what I have to say is, just over the last decade or so, what has been really quite fulfilling is that the field has really advanced in a lot of these areas that I've been studying, whether it be salivary cancers and thyroid cancers, particularly in our biologic understanding of these diseases, the genomic landscape of these diseases. And then along with that, really the development of targeted approaches that allow you to ask important questions directed at those molecular targets. And that has really yielded a really nice developments in the field with a number of different targeted agents that have been approved for rare cancer indications in the head and neck, and really provides some promise and motivation to continue to reach for these therapies and conduct these trials. So, you know, when thinking about the way that rare cancer indications come about, of course, there are very two obvious ways that these occur. And the first, I'm hoping you can see my pointer here, is the investigation of these targeted therapies against multiple biomarker-selected cancer subtypes, such as in basket trials. And then in those basket trials, finding a specific indication in a rare cancer, and that leads to that indication. And of course, the second way that this may be developed is someone coming in and being focused on a specific rare cancer subtype, looking at the biology of those diseases and designing clinical trials specifically for those diseases. And of course, there's a mixture of those in between. And so today, I'm going to really be sharing two general topics or stories. In the first half, I'll be talking about the development of this drug, Tipifarnib, for HRAS mutant head and neck squamous cell carcinomas. And this really developed in that first way, where we were looking at it in a basket study setting, and then finding the specific signal in head and neck squamous cell carcinomas. And what's been very important for how we've conducted business here in rare cancer therapies is really strong partnerships with basic scientists and translational scientists. And for this, you know, Jim Fagan, who's the head of the medical services here at Memorial Sloan Kettering, and previously the head of endocrinology, and his translational group, of which Brian Unch, who's also a head and neck surgical oncologist and a member of his laboratory, their science has really kind of helped drive that clinical trial program. And in the second half of the talk, I'll be talking about our attempts at building a salivary gland cancer program here at Memorial, and then also a lot of the exciting ongoing work in salivary gland therapeutics. And for us, the translational part of that, the basic science part of that, has come from Luke Morris, who again is another head and neck surgeon here, who with his own laboratory efforts and his interest in genomics and immunogenomics, that's really helped drive the biologic rationale for our trials. So just going into that first topic, this is really a story about bringing back farnesyltransferase inhibitors, or FTIs, back into the clinic. So as many of you in the audience probably are well aware, well over more than a decade ago, there was an interest in developing FTIs as a targeted strategy for RAS mutant malignancies. And this was really based upon the understanding that all three isoforms of RAS, even when mutated, require localization to the membrane for activation of its oncogenic downstream signals. And this occurred by a process called prenylation, which is basically the covalent attachment of a hydrophobic group to the C-terminal tail at this CAX motif, the cysteine at the position 186. And so in human cells, this is primarily the prenylation event that predominates is farnesylation, which is catalyzed by the farnesyltransferase. And this is what this in vitro data figure illustrates, is that both KRAS, NRAS, and HRAS are all susceptible to farnesylation. And it's primarily the addition of farnesyl groups to the C-terminal tail that leads to this hydrophobic group and allows attachment to the membrane, localization, and downstream signaling. And so the rationale here is, in order to disrupt this process, you could develop a class of drugs that inhibits farnesyltransferase, known as FTIs, that would prevent the farnesylation of RAS, prevent its localization to the membrane, and thereby disrupt its oncogenic signaling. And in RAS mutant malignancies, this would be a way this would translate to a very effective anti-tumor effect. So a number of different trials were done with several different FTIs, and while there were some signals in those studies, ultimately nothing was brought to bear in terms of significant clinical activity that led to FDA approval in any indications. And one of the biologic reasons for this was in the context of farnesyltransferase inhibition. In fact, there are other redundant prenylation events that become more relevant that allow for the addition of another hydrophobic group to these RAS, these mutant RAS, that allows persistent membrane localization. And one of the predominant forms of this is geranylation by geranylgeratylases. And so this is an experiment demonstrating this concept where in cells with radiolabeled cells and using HPLC, you could see that with increasing doses of the shearing plow FTI, you get decreased farnesylation of RAS, but you concomitantly get increased geranylgeratylation. And so the idea here is that the failure to find a significant signal the first time around for FTIs was really because RAS was not being properly delocalized and signaling wasn't properly being abrogated in those tumors. So from that same paper back in 1970, this is a Schering-Plough paper, just to demonstrate why, however, FTIs may have particular significance in HRAS mutant malignancies as opposed to KRAS and NRAS. So here again are these HPLC experiments where in untreated KRAS proteins, you find predominant farnesylation and less granulation. But then in exposure with FTIs, you decrease farnesylation and these KRAS proteins then are granulated. However, when you look at HRAS proteins, again, you see the predominant farnesylation, but in the context of FTIs, you don't get the commensurate granulation. And indeed, what several studies demonstrated in the late 90s was the fact that unlike NRAS and KRAS, HRAS was not susceptible to these redundant prenylation events, and this would predict that they would be more susceptible to FTIs. And so Jim Fagan's group, as I mentioned in the beginning, took on this question a little bit more recently, looking at how FTIs worked in a variety of different in vitro cell lines with HRAS, NRAS, and KRAS mutations. The reason why this was a question of importance for that group is, Jim's group really focuses on thyroid cancer development therapeutics and biology. And thyroid cancer is one of the rare malignancies where all three RAS isoforms can be found mutated. And so this is work from Shi Chen, who was a postdoctoral fellow in his group. And here's one of the first experiments he did was subcellular localization of different RAS isoforms exposed to FTIs. This happens to be an HRAS mutant thyroid cancer cell line. And what he was able to demonstrate is that at baseline, all three RAS isoforms are really localized to the membrane. But when then exposed to an FTI, the HRAS then delocalizes from the membrane into the cytosol as one would predict. But then NRAS and KRAS, because of these redundant prenylation events, remain localized to the membrane. So again, illustrating this principle that more than a decade previously had been demonstrated in vitro as well. And then when you look at FTI effects over time in this HRAS mutant thyroid cancer cell line, 2 hours up to 72 hours, you see the slower migrating form of HRAS arising, which really represents this unfarnesylated form, and then concomitant inhibition of downstream signaling as illustrated by decreased phospho-MEC levels and increased induction of apoptosis, which then translates to pretty good anti-proliferative activity and proliferation assays across different histologies. These are cell lines that represent melanomas, bladder, breast, and thyroid cancers, all HRAS mutant. And when you compare it to the anti-proliferative effects in NRAS and KRAS, clearly the IC50s are much lower in the HRAS mutant population. So this formed the basis, again, demonstrating that FTIs, while maybe not as effective in the previous earlier trials where there wasn't genomic selection, that perhaps a strategy for selecting for HRAS mutant disease would be the appropriate way to find a better clinical efficacy signal. So this was taken on from the in vitro setting into the in vivo setting by Brian Unch, who's the head and neck surgical oncologist in Jim's group, where he was evaluating FTIs in a genetically engineered mouse model that he developed of thyroid cancer, which had a knock-in of a mutant HRAS, a G12V allele, and a P53 knockout. And this genetically engineered mouse model creates very aggressive thyroid cancers, specifically a mix of poorly differentiated and anaplastic thyroid cancers. So anaplastic thyroid cancers being completely undifferentiated disease that leads to very aggressive tumors and often mortality. So in this setting, when he treated these animals with Tipifarnib, the first-in-class farnesyltransferase inhibitor that was developed, you can see that for the majority of these animals, tumor growth was actually inhibited, or even tumor regressions were seen. And he could correlate this to the appearance of, again, unfarnesylated forms of HRAS arising and inhibition of downstream signaling by phospho-MEC and phospho-ERK. So again, an in vivo demonstration of this in an aggressive thyroid cancer mouse model. So all of this, again, formed the basis for us wanting to test this idea clinically, and Cure Oncology became the partner in this, buying the rights for Tipifarnib from J&J, which was, again, the first-in-class FTI that was developed. The way that Tipifarnib works is that it binds at the CAC site in RAS and blocks the ability of RAS to be farnesylated at that site. And again, Tipifarnib specifically had been studied in over 70 different studies and more than 5,000 patients, both in solid and hematologic malignancies. While there were promising signals there, again, none of the signals reached the level for FDA registration for any specific indication. And when you look at its in vitro farnesyltransferase inhibitory activity, it's quite potent against lamin B or KRAS in the nanomolar range. So the basket study, that concept that we helped develop, was really a quite simple one, which was taking 2 cohorts of patients. One cohort dedicated just simply to thyroid cancer because of the preclinical data we had developed in Dr. Fagan's group. HRAS mutations can occur actually both in follicular cell-derived thyroid cancers as well as mesothelioma thyroid cancers. And then in cohort 2, just looking at its effect in HRAS mutant other solid tumors, so any other solid tumor with HRAS mutation. Again, we know that these are kind of rare alterations to be seen in human malignancies, and the feasibility of moving forward with this in this biomarker selected way wasn't clear at the time. So each of these cohorts were going to be evaluated with a primary endpoint of objective response rate in a 2-stage design, each assessed individually. The first stage, we would require at least 2 responses in the first 11 patients to move on to the second stage, for a total of 18 patients if you move down to the second stage, and really targeting a 30% overall response rate, which would require seeing at least 4 responses. And of course, we'd be looking at PFS or duration of response and safety and tolerability. So when the first stage was completed, surprisingly to us, we did not progress to the second stage with the thyroid cancer cohort. And in the first stage, we had seen 2 responses. And both those responses occurred in head and neck cancer patients out of 3 that we accrued to that first stage. We had also accrued 5 salivary cancer patients, which I'll get to in a second, further in the talk, and then 3 other histologies. But because of this specific signal that we saw in head and neck cancer, the decision was made that when we opened the second stage, dedicated to further exploring that head and neck cancer signal. And so this is how, over time, the trial design evolved to really react in real time to data that we were seeing on the clinical trial. And this is how we changed that design. So cohort 2, that other solid tumor cohort, really became a head and neck squamous cell carcinoma cohort. And then a third cohort was also added, thinking that perhaps there was something to Tipifarnib efficacy in HRAS mutant squamous cell cancer lineages beyond head and neck, and wanting to test that idea in the third cohort. So as we kind of accrued more head and neck cancer patients to the trial, by the time we had accrued 16 head and neck cancer patients, in addition to 4 other squamous cell cancer histologies, an interim analysis was performed to really look at where were the responses we were seeing. And one of the analysis that was run was associating response to varying allele frequency. And what was discovered is that almost all the major responses depicted by these dark blue bars were seen in those with high variant allele frequency, defined as 35% or greater of HRAS mutations. And you could encompass all partial responses if you then broaden this out to greater than 20% allele frequency for the HRAS mutations you were detecting. While lower than that, a lot of the patients were inevaluable or had progression of disease as best overall response. And so while this was a small sample, with really the desire to get this right the first time around and employ the best strategy for finding the largest signal in this disease, the protocol was subsequently amended to the last version, which is depicted here. Again, looking at cohort 2, dedicating it to head and neck cancer patients, now up to 30 patients for that cohort, but limiting enrollment to those with high variant allele frequency. So greater than 20% if you had a serum albumin of 3.5 grams per deciliter or higher, or just 35% or greater, regardless of albumin. The other modification that was made is that the initial dose and schedule of Tipifarnib used was 900 milligrams twice daily for 1 week on and 1 week off for 4-week cycles. When looking at the data, it was pretty clear that a majority of patients were dose reducing by the end of the first month down to 600 milligrams twice daily. And so we also altered the dosing on the trial in the interim to make this more tolerable. So at ASCO this year, we presented the results of the high variant allele frequency head and neck cancer patients. So out of the 30 patients we accrued to that cohort, 21 met that definition of high variant allele frequency. At the time, at the data cutoff for ASCO, We had 20 patients treated at that time and 18 who were evaluable for efficacy. One patient was not evaluable because it had not started treatment yet, and a second had withdrawn from the study prior to the first evaluation. So the median age for these patients was 64. The majority of patients were men. You can see the distribution of the primary sites of tumors. The majority of patients had oral cavity cancers, which is consistent with previous genomic studies for head and neck cancer patients where HRAS mutations are found. This was a heavily pretreated patient population with a median of 2 prior anti-cancer treatments. As you can see that almost all patients had received a platinum previously, almost approaching two-thirds had immunotherapy prior to trial enrollment, and about half had been treated with the EGFR targeting antibody cetuximab. So here's the bottom line efficacy data. So depending upon whether you did a modified intention to treat analysis of all 21 high variant allele frequency patients, the response rate is about 43%. If you just included the 17 patients who were response available, that's 47%. The 18 here is the one additional patient that we included in the analysis was treated on an SPU using the same protocol. And if you included that patient, it was 50%. But overall, very high response rate for a targeted agent in this disease, which it has to be mentioned that the only targeted therapy that's really been approved for head and neck cancer has been cetuximab, or the EGFR targeting antibody, with a long median duration of response of 14.7 months. And the responses that we're seeing on the trial were quite dramatic. This was one of the early responders from the Dana-Farber, a 59-year-old woman with nasopharyngeal squamous cell carcinoma who had progressed on cisplatin and previous pembrolizumab, with a dramatic reduction in this nasopharyngeal tumor. This patient had an HRAS G12D mutation. This is a patient that was treated here, a memorial 55-year-old man with metastatic laryngeal squamous cell carcinoma at multiple sites, treated with cetuximab, Taxol, pembrolizumab, and with palliative RT, and at the first radiographic assessment had a 22% overall reduction, including this reduction of the subcarinal mass that was seen. And lastly, this was a patient, a 69-year-old man with recurrent oral cavity squamous cell carcinoma who had just rapidly progressed off a combination of nivolumab and another investigational immunotherapy. This patient did have both a P53 mutation and an HRAS G12S, with a dramatic and remarkable reduction in this head and neck tumor, filling up the sinuses with visible resolution at the skin. So also presented at ASCO this year was a comparison of the median PFSs we saw between Tipifarnib treatment and the prior systemic therapy that these patients had received. So as opposed to having achieved 1 PR and 6 stable disease responses in those that we had documentation of this, and a median PFS of 2.8 months with prior therapy, among the 9 PRs and 9 stable disease that we saw with the Tipifarnib treatment, we saw a median PFS of 5.9 months. So in a later line of systemic treatment, an improved progression-free survival when compared to last therapy. The overall survival also was quite long here, a median overall survival of 15.4 months for this cohort, which compares favorably to overall head and neck cancers historically. What we don't know and what we really need to investigate is how this compares to historical HRAS mutant survival, and this is one of the thrusts of ongoing investigation. So, our conclusions from this basket study was that we saw encouraging anti-tumor activity of Tipifarnib in recurrent metastatic head and neck cancers with high HRAS mutant variant allele frequencies. The response rates, depending upon what you put in the denominator, was 40 to 50%. The duration responses were quite long. The median PFS was favorable when compared to the median PFS of last systemic therapy for these patients, and a pretty impressive median overall survival, but we really need that reference of HRAS mutant head and neck cancer patients to understand the meaning of that. And this response activity was seen despite resistance to prior chemotherapy, immunotherapy, or cetuximab. And based upon this promising signal, ongoing currently is an international registration trial, which is also open at your center, looking at a Tipifarnib in head and neck squamous cell carcinoma patients with high HRAS mutant variant allele frequency. It's a single-arm phase 2 trial, really looking to lead to registration for the drug in this specific genomic cohort of head and neck cancer patients. The registration therapeutic trial is known as AIM-HN, where HRAS mutant head and neck cancer patients are then being treated with Tipifarnib. The primary endpoint here is objective response rate. The total sample size is 59 with high variant allele frequency HRAS mutations. We are also allowing a cohort of patients to be treated with low variant allele frequency to further thresh out whether there are differences by variant allele frequency. So you really only need HRAS mutations to be on the trial. And the secondary endpoints that are listed here for both to be evaluated both in the high variant allele frequency and the all variant allele frequency cohorts. In parallel in this trial, we have SEEK-HN, which is really an observational study, which I was referencing before. We're accruing both the HRAS mutant patients that are being treated on the clinical trial and comparing it to wild type controls to really better understand the clinical and demographic features of the HRAS mutant head and neck cancer cohort, as well as their responses to other therapies in the first line setting. So we're hoping to get out of this study not only the efficacy of Tipifarnib, but a better understanding of what HRAS mutant head and neck cancers look like. Beyond head and neck cancer, there have also been trials done in other HRAS mutant histologies. This was a urothelial carcinoma study that was conducted by Dr. Se-Hoon Park at Samsung Medical Center in South Korea and presented at ASCO this year as well. As you may know, RAS mutations occur in about 11% of urothelial carcinomas, over half of which are HRAS mutations. And in fact, in the first generation of these trials, Tipifarnib was actually evaluated in a phase 2 urothelial carcinoma study without genomic selection, and at that time showed only a 6% overall response rate. But this was a study to look at it in HRAS mutant urothelial carcinoma, similar to what we had been doing in head and neck cancers. Now, in this trial, they not only selected for HRAS mutations, but also for patients with single nucleotide polymorphism and STK11, which this group had previously found to correlate to poor prognosis. So they accrued both those types of patients, in total treated 21 patients, and compared to that 6% overall response rate in the unselected trial, they observed a 24% response rate with a median progression-free survival of 4.7 months. And here's the waterfall plot from that study. What's really noteworthy here is that all the regressions and responses were seen in those with HRAS mutations, and the wild type group, the STK11 SNP group, which are depicted by these yellow bars, did not respond at all. So this is, again, direct clinical evidence demonstrating that really benefit of the therapy was relegated to those with HRAS mutations. As I mentioned in the first basket trial, we had also accrued a good number of salivary gland cancer patients. Now, this is a diverse set of diseases, which I'm going to dedicate the second half of the talk discussing, but it's a diverse array of histologies, and depending upon the histologies, HRAS mutations actually can be quite common, such as myoepithelial carcinomas, where it's up to 20% of those patients. And so we had accrued 7 salivary gland cancer patients, the second cohort of the clinical trial. 6 additional ones were treated with Tipifarnib off protocol through expanded access programs. And Dr. Glenn Hanna at Dana-Farber was really interested in gathering all of this data together in this analysis, which we did together, which was published in Cancer earlier this year. So out of the 13 salivary gland cancer patients, 12 had resist evaluable disease. And in this group, we only saw 1 major response out of those 12, with a median progression-free survival of 7 months and an overall median overall survival of 18 months. And here are the different histologies, the most common of which was salivary duct carcinomas and myoepithelial epithelial carcinomas. We'll be talking about these a little bit more later. And here in the waterfall plot, what you can see is that 6 out of those salivary gland cancer patients achieved some type of regression. And when looking at the genomics, P53 mutations were common in this group, but also PIK3CA mutations that can signal in a parallel fashion to the RAS-MAP kinase pathway and could conceivably mitigate efficacy, although in this small sample, of course, it's quite hard to demonstrate its real effect on efficacy here. So when looking at the swimmer's plot, just want to point out that the one responder was quite durable, which was ongoing at the time of ASCO presentation of 14 months, and 7 patients out of the 12 that were resist evaluable had a best response of stable disease with a median duration of disease stability of 9 months. So even though we didn't get a lot of major responses here, there was pretty significant stable disease among these patients. But clearly what the head and neck, the bladder, and the salivary gland cancer experience demonstrates was that even though all three studies were done with HRAS mutant disease, there was a wide variance in the degree of efficacy seen. And so clearly there is some biologic context or lineage-specific effects that are influencing Tipifarnib efficacy. So when thinking about those different contexts, it's important to note that in head and neck, HRAS mutations actually define a pretty unique biologic molecular head and neck cancer subset. And so when we're doing that SEEK-HN study, we're really trying to understand how that unique biologic subset translates into clinical phenotypes. And in the TCGA study, HRAS mutant subsets of head and neck cancers were distinct in that they were characterized by low rates of copy number alterations, less frequent caspase-8 inactivation, and less frequent p53 mutations, although even in the limited case the examples I showed you, some of the patients we enrolled on the trial had p53 mutations. And interestingly, just like in colorectal cancer, you can see the emergence of RAS mutations in patients who are resistant to cetuximab, and there is indeed evidence that you can see HRAS mutations emerge in liquid tumor biopsies, and those are resistant to cetuximab in head and neck cancer as well. So, but in thinking about, well, what are the biologic parameters here that could be determining efficacy? I go back to Brian Unch's work in the thyroid cancer models and those thyroid cancer genetically engineered mouse models, where again, he saw pretty good inhibition of tumor growth or even regression in those tumors. He also found that when you looked at Tipifarnib effects in vitro in HRAS mutant cell lines, you do get inhibition of the pathway, but you do commonly also see with increasing concentrations of Tipifarnib, increased NRAS and KRAS GTP loading, indicating that while you could inhibit HRAS mutant signaling, you are probably mitigating your ability to inhibit pathway activation because of increased signaling through NRAS and KRAS that are probably occurring through RTKs. So this adaptive resistance is probably mitigating the full therapeutic potential of FTIs in thyroid cancer tumors as well. And so the question is, these lineage-specific differences we're seeing in thyroid or bladder and salivary versus head and neck, is it due to differences in these adaptive responses to FTI and FTIs? And that's something that we really need to investigate. The other thing Brian was also able to delineate was that when he treated his mouse models for prolonged periods of time, he could also observe acquired resistance. And in this example, in these three mice that developed acquired resistance to Tipifarnib, he sequenced all three, and in two of them he found fairly interesting findings. One was the emergence of an inactivating NF1 mutation, which was really important and a key observation, because NF1 is a tumor suppressor that can inhibit RAS MAP kinase signaling. And by having a deleterious and correlating resistance to a deleterious mutation in NF1 really validates that the true target of Tipifarnib in these models was RAS MAP kinase signaling. And the second was a hotspot mutation in GNAS, which is the alpha subunit of a heterotrimeric G protein complex that activates adenyl cyclase, increases CAMP and protein kinase signaling, which in thyroid cancer has a particularly meaningful role in activating differentiation and proliferation and growth in parallel to RAS MAP kinase. So in addition to demonstrating that acquired resistance can occur, and we're certainly seeing that in clinically as well, the specific genetic lesions found, at least in the thyroid cancer mouse models, suggest, again, validating that that Tipifarnib is really targeting the RAS MAP kinase pathway, at least in those models. And the question then also becomes, well, clinically, the differences that we're seeing in Tipifarnib activity, how does that correlate to the different types of acquired resistance that are being seen in different tumor lineages? So the outstanding questions that we're wrestling with and we really want to delve into, beyond trying to establish the signal of activity that we saw in head and neck cancer, is what are these lineage differences? What are the biologic basis of this? Do they have to do with the genomic alterations that are also seen concomitantly with HRAS mutations in these different tumors? Are there different biochemical adaptive responses that we're seeing? Are the adaptive responses that Brian was able to illustrate in thyroid cancer models relevant for other tumor types? Are there other farnesylated targets within these tumors that are also contributing either to resistance or to susceptibility is an open question. We've been investigating Tipifarnib in HRAS mutant disease, but there's a higher percentage of HRAS amplification that can also be seen in human malignancies. Is there activity against those patients? And of course, the idea of combinations. I mentioned the emergence of HRAS mutations is cetuximab resistant. Could you combine this with cetuximab or other type of agents to boost efficacy? And so these are things that really need to be investigated both preclinically and then brought to the clinic. So that's the first half of the talk, really talking about FTIs and the head and neck cancer signal. And usually I would take questions at this point, but I know we're taking questions at the end, so please feel free to ask me questions at the end about that first half of the talk. So the second half of the talk is really talking about salivary gland cancers. And as I mentioned, very rare disease, no FDA-approved agents, no standard therapies for these patients. And when we talk about salivary cancers, I just also wanted to highlight, we're not only talking about cancers that arise in the three major salivary glands, the parotid, the submandibular, and sublingual glands, but all through the upper aerodigestive tract, we have microscopic salivary glands as well. So sometimes when you'll see a base of tongue tumor or cavity tumor that you assume is squamous cell carcinoma, or something in the sinuses that you assume is squamous cell carcinoma, and the biopsy comes back as adenocarcinoma or adenoid cystic, what that illustrates is really that those are tumors arising from those microscopic minor salivary glands. So back in 2017, in the 4th edition of the WHO classification, this is now the listing of different histologies in salivary cancer. So heterogeneous in terms of clinical, histologic, and biologic types of salivary gland cancers. What was really important in this reclassification is the inclusion of pathognomonic genetic alterations in these tumors, including mutant HRAS, again, for epithelial and myoepithelial carcinomas. But just as a sidetrack, I just want to highlight secretory carcinomas with the ETV6 NTRK3 rearrangements, because of course this is relevant for therapeutics and the recent approval for TRK inhibitors. So going back to the development of these NTRK inhibitors, specifically larotrectinib, and NTRK fusion positive tumor types, when looking at those trials that led to the approval of larotrectinib for TRK rearranged malignancies, you can see actually the majority of the cancer subtypes that were treated on those initial trials were salivary gland cancers. And these were really these secretory carcinomas that are characterized by ETV6 NTRK3 rearrangements, previously known as mammary analog secretory carcinomas. And in the waterfall plots of the tremendous therapeutic window seen with these, with larotrectinib, in purple are all of the salivary gland cancer patients that you can see responding extremely well to this particular targeted therapy. So this is again as an example of a large multi-malignancy type of biomarker-driven basket study or cohort study, where the signal was delineated in this rare cancer indication and led to an approval that benefited those rare cancer patients. So when we had been involved in this story early on, I had this particular patient that we published on, treated with crizotinib because it was at the time before these TRK inhibitors were widely available, had weak TRK activity and seen some responses in this NTRK3 rearranged secretory carcinoma patients, and then was enrolled on the entrectinib trial, which is from Ignita, again also approved for TRK rearranged malignancies and can be used in these patients. We saw the emergence, along with acquired resistance, the emergence of this NTRK3 G623R mutation, which is a solvent front mutation that competes off binding of the drug and in vitro leads to a lack of inhibition of the appropriate pathways for drug inhibition. And so similar to the developmental story for ALK, ROS, et cetera, there have been now, of course, the development of second-generation TRK inhibitors as well that have benefited these patients. So this is one patient that Alex Drillin, who leads the TRK program here at Memorial, and I had treated with this very invasive secretory carcinoma with a TRK3 rearrangement. Initial response with entrectinib that then developed acquired resistance and the G623E mutation that then responded to a second-generation drug from TP Therapeutics, repotrectinib. So again, it's following down along the same paradigm as these other targeted therapies, and how second-generation and perhaps third-generation drug can overcome resistance seen with those inhibitors. But going back to kind of the salivary programs that we've been developing here at Memorial or trying to develop, they've really been focused on the most prevalent subtypes. First, adenoid cystic carcinomas, which we'll speak on in a second, and secondly is salivary duct carcinomas. So these are the most common that we see as medical oncologists, but widely different biologically and clinically. And biologically, we have at our disposal really pretty good sense of the genomic landscape of these diseases. And when you compare ACCs to salivary ducts or SDCs, immediately you see the differences, which is very low mutation burden among ACCs and a higher one among salivary duct carcinomas. And many of the mutations that we see in ACC are really not readily targetable, while as over 60% of salivary duct carcinomas will have some kind of mutation that is targetable. So again, a rare disease in salivary duct carcinomas, but a lot of therapeutic opportunities and research opportunities to better understand this disease and how these targets can be exploited. But we started with ACC when I first came onto the service, and one of the first things we did was we partnered with Scott Laurie's long-term effort to really understand how the efficacy of systemic therapy in these diseases. And when you look over from 1966 to 2009, one of the issues that comes out is that these patients were understudied. So if you look at phase 2 data, you have less than 500 ACC patients that were studied. And ultimately, this is thought to be a chemotherapy insensitive disease with about 10% or less single agent activity with vinorelbine, platinum, or anthracyclines. I still use chemotherapy for these patients, but what these clinical trials demonstrated was that we needed more research and we needed better ideas for these patients. But the problem was, there was an intellectual gap because we didn't really at the beginning understand the biology of these tumors. And one of the big breaks was in 2009, Gordon Stemman's group published the identification of a unique translocation, 6-9 translocation that characterizes over half of ACCs, which creates a fusion protein between MIB and NFIB and leads to the overexpression of MIB. And it turns out that MIB is an oncogenic transcription factor that is found to be implicated in T-cell ALL, and its amplification found to be implicated in colon and breast cancer oncogenic phenotypes. And so the idea here was that one of the drivers for ACC was the overexpression of this oncogenic transcription factor MIB. The problem, of course, is that it's not an enzyme or something that's readily targetable by a drug. But the idea is, could we learn more about the biology of MIB and design clinical trials based upon that? And one of the first ideas was VEGFR multi-tyrosine kinase TKIs, such as exitinib. And the reason for this is that some of the downstream transcriptional targets of MIB include VEGFR, PDGFR, and KIT, which are the targets of these drugs. And so one of the first phase 2 trials we did in ACC was a phase 2 trial of exitinib, which ultimately was a negative study because we only saw 3 major responses in the green here. But you can see the majority of patients had tumor regressions, and for a chemo-insensitive disease, not a bad result. And ultimately found a 9% confirmed response rate. Over 2/3 had some degree of tumor shrinkage, and some were on it for quite a long period of time. A subset of these patients underwent next generation sequencing. The most common mutation here was a Notch1 mutation, which I'm going to get to in a second. But we also saw 4q12 amplifications. And the reason why this amplicon is of interest is because this is the amplicon that involves PDGFR, KDR, and KIT. And indeed, these patients had higher expression of all these genes that were targeted by exitinib. And it turned out that these 3 patients did have outsized clinical benefit to exitinib that hits these targets, such as in this patient with lung ACC with a 14-fold amplification at 4q12. We went on to investigate regorafenib in the phase 2 setting. And again, we didn't see major responses with this drug. The idea here was that this hit many of the same targets as exitinib, but also FGFR. But what we found in the swimmer, as depicted in the swimmer plot, is that these 4q12 patients again seem to have an outsized clinical benefit, with 2 out of the 3 patients who were on trial the longest having pretty significant 9-fold amplifications for 4q12. The one patient that was on it for only 2 weeks came off for toxicity and in fact had a 14% regression after only 2 weeks of therapy. So indicating that in ACC, those with 4q12, that this approach of VEGFR TKIs may be quite favorable. And what we also found in that study is that similar to what you see in thyroid cancer and renal cell carcinomas and hepatocellular carcinomas, because these are not, the targets of these drugs are not completely redundant, you can see efficacy sometimes even when they've progressed on previous TKIs, with stable disease and tumor regressions seen in patients that have progressed on axitinib, davitinib, et cetera. The last TKI study that we've done is lenvatinib, and this was published last year in the JCO, and by far it was our first positive study in ACC. By far had the best clinical signal with a 15% confirmed response rate and a prolonged median progression-free survival, as you can see with the swimmers plots. And by these scans, you can see that many of us, we did see some dramatic regressions in these patients, including the appearance of the necrotic centers in a lot of these tumors, characteristics of these TKIs. But of course, you have to balance it with toxicity, and lenvatinib being approved in renal cell, hepatocellular, and thyroid cancer, we saw similar rates of grade 3, grade 4 toxicities as was seen on those prior trials, but still this was an issue in terms of 18 patients on the trial were having to stop lenvatinib for some toxicity related reason. And so balancing both the safety and the benefit of, for ACC patients and all patients with these TKIs is going to be critically important. So there have been a number of these TKI trials now been done. Lisa Lasitra and Laura Licati in Italy had also done a lenvatinib trial demonstrating pretty good response rates. But there's an ongoing study with a drug that was previously known as ipatinib when it was tested in a phase 2 trial in China that demonstrated a 47% major response rate. So there's an ongoing trial with this drug now with Elevar Therapeutics under the name rivoceranib. And of course, if that study approaches any of the response rates seen in China, this would be by far the best option. So we anxiously await the results of that ongoing trial here in the US and Korea. So ultimately, there's activity with TKIs, but we have to manage toxicities. It's not dependent upon MIB status. We did those correlations ultimately, and you can't really see a good relationship between MIB IHC status and TKI activity. And 4Q12 amplified tumors may have particular benefit. Well, with regards to Notch mutations in ACC, Renato Ferrato and the group at MD Anderson were the first to demonstrate that activating Notch mutations, these are alterations in the PES domain or the negative regulatory region, really correlate to a different type of clinical subset of ACC patients. These are very aggressive tumors with poor recurrence-free survivals and a distinct pattern of metastasis, particularly to the liver, bone, and other sites. Luke Morris and Alan Ho at Cedars-Sinai recently published in the JCI last year in an assessment of over 1,000 ACC cases, again demonstrating that Notch1 mutations really speak to patients with a much worse survival, much more aggressive disease. So while there aren't a lot of targetable alterations in ACC, Notch1 mutations in recurrent metastatic disease setting can occur in up to 20% of recurrent metastatic ACC patients. And so if you were to design a targeted therapy against a genomic alteration, activating Notch mutations would really be it. And so Allele Pharmaceuticals took on this question by testing a gamma secretase inhibitors known as AL101 in Notch1 mutant, activating Notch mutant ACC patients. And so the way gamma secretase inhibitors work is that while you have activating mutations in Notch, they still are dependent on gamma secretase activity to create the intracellular domain that's activated within tumor cells. And so by inhibiting this enzymatic step, you could still inhibit downstream signaling in Notch mutant tumors. And so this question is being taken up on an ongoing study, and these are results that were presented by Renato Ferrato, who's the PI of this trial at this year's ESMO, and reporting on results of cohort 1, which were Notch activated Notch mutant ACC patients that were treated with AL101 and a 4 milligrams for every week weekly dosing, with a primary endpoint of overall response rate. The ongoing trial is enrolling to the second cohort, which is giving AL101 at a 6 milligram weekly dosing. And what she presented there was this data, which was a major response rate of about 15% in this cohort, and a disease control rate of 68%. And again, given the aggressiveness of this disease compared to wild-type ACCs, this is actually quite a promising activity and minimally demonstrates that activating notch alterations are a real driver oncogenic event in these tumors and a relevant therapeutic target. And indeed, some of the responses that were observed on the trial to date are really quite impressive, such as this patient with disease loaded in the tumor with a dramatic regression in response, and another patient here with liver disease. And you can see all these examples of really impressive regressions. And so again, demonstrating this is a very good proof of principle that this is a relevant therapeutic target for this disease. And hopefully similar and better results will also be seen with the 6 milligram weekly dosing that's currently being tested. So for such a small disease, when we started more than a decade ago and TKIs were really the only game in town, it's really amazing to see how much clinical trial activity is occurring in ACC specifically. So as I spoke about, there's ongoing evaluation for Zarnestra as hopefully a next generation TKI that's going to be even better than lenvatinib that we tested. There are a variety of mid-targeted approaches. As I mentioned, this has been hypothesized to be the central oncogenic driver with an all-transparent acid study being done at Dana-Farber to suppress MYB expression. There's also a MYB vaccine approach that's being tested in Australia at Peter Mac. The genome-directed targets such as notch, targeting notch activation, and then there's an MDM2 targeting program in Michigan. The vast majority of ACCs are p53 wild type, which justifies that approach. There have been several epigenetic studies that have been done and some activity seen with vorinostat, the histone deacetylase inhibitor vorinostat, with a trial ongoing in China, and an arginine methyltransferase inhibitor PRMT5 that's being investigated by Prelude Therapeutics and as well as GSK, and a variety of different immunotherapy approaches that we are investigating, as well as in combination with VEGFR TKIs and chemotherapy and radiation. And also there's a literature demonstrating that PSMA is widely expressed in adenoid carcinomas, and whether radio-labeled approaches targeting PSMA would be relevant is being investigated. So I had a lot more in terms of salivary cancer studies and salivary duct carcinomas, but I think looking at the time, we're kind of running out of time. So I'm just going to end it there, and I'm just going to skip ahead to our acknowledgements and perhaps just open it up to questions at this point. So in terms of acknowledgements, you know, a lot of what we've done here at Memorial is obviously centered on the head and neck medical oncology service, but a lot of collaboration has occurred with our with our DMT, both on the preclinical side and the clinical side. I just want to further identify Dr. Fagan and Brian Unch for their work on the FTI story, Luke Morris and Alan Ho, who's been really instrumental in helping with the ACC program and some of the salivary duct studies that I won't be able to share with you today, and of course our NIH support. So with that, I'm happy to take any questions.
Thank you very much, Dr. Ho, for this excellent presentation. I want to remind the attendees, if you have any questions, to please use the Q&A feature located at the bottom of the screen. I think we have one question from Dr. Shin, which states, do you have direct biologic evidence that Tipifarnib can be synergistic with an EGFR inhibitor?
Those studies are those preclinical studies are ongoing, actually. So yes, you know, you would predict based upon how RAS mutations can mediate resistance to cetuximab in the colorectal setting and the emergence of these mutations in head and neck that would be the case, but the actual data is pending. No, we don't have that yet.
Yeah, actually, we did do some studies here, non-published yet, Alan, which basically we looked at Tipifarnib in non-HRAS mutated head neck cancer, and we did see some synergy with cetuximab.
Oh, great.
With the HRAS mutation, that changes basically the equation, of course.
Yeah.
So we're working on that as well.
Oh, that sounds promising.
It makes sense. I want to ask you, you know, while we're waiting for questions, so the TP53 story with HRAS, do you believe I mean, the TCGA data is very impressive in terms of these patients. While you have TP53, those are not mutually inclusive. But as you said, you had one patient who had both. What do you believe is going on there? Do you believe it is mutually?
I think one of the things that we have to do with the ongoing patients that we're treating on the Tipifarnib trial and on that observational cohort is to kind of get at these questions, because certainly the TCGA has been enormously helpful for developing these trials and better understanding these diseases. But those are earlier stage tumors as well. And I mean, similar to the Notch mutant story in ACC, which is just one example of many, you know, in the beginning, when we were looking at the genomic landscape of Notch mutations in ACC, they were, you know, less than 5%, 6%. But that was primarily because those were done on surgical series. And in fact, there's a huge enrichment for Notch mutations when you look at recurrent metastatic tumors in the 20 to 25% range. So similar to HRAS mutations, their frequency as well as the frequency of their co-occurring mutations, what exists in early surgical, in you know, primary surgical specimens versus those with recurrent metastatic disease still remains to be determined. And we've seen a number of patients with P53 mutations. So they're definitely not mutually exclusive. And I guess it's possible that there's an enrichment for more P53 in the recurrent metastatic head and neck cancer patients that we're treating on the trial. We just have to look and see.
Great. I have a question from Andreas Heilman, which states, thank you for the wonderful seminar. What would be a good agents to combine the gamma secretase inhibitors with a Notch1 mutated ACC? I think you touched a little bit on that, but maybe. Yeah,
you know, those kind of ideas are currently being explored. You know, one of the initial thoughts was really how this could be combined with MIB targeting. You know, Brad Bernstein's group in Boston had this beautiful paper a couple of years ago, really demonstrating that there are really two arms of signaling in ACC, those that are MIB directed and those that are Notch directed, and really kind of dividing those up by the myoepithelial components of the tumor as well as the ductal components of the tumor. And so, you know, and then looking at Luke Morris and Alan Ho's JCI paper looking at the genomic landscape in over 1,000 ACC patients, they had categorized different mutation subtypes or subsets of the disease, and one of them was Notch mutant and MIB rearranged. And so you wonder that whether or not targeting MIB effectively in combination with Notch would be a really interesting approach. The barrier to that is really finding ways to target MIB effectively, and you know, we currently don't have that drug. And you know, Glenn Hanna's study at Dana-Farber looking at all-transformernonic acid as an attempt to do that is going to provide some insights. We had tried to do that with AKT inhibitors, actually, in a trial that I didn't have time to mention, with the Merck AKT inhibitor. That didn't demonstrate an interesting clinical signal, but did show that we could downregulate MIB. So, you know, I think teleologically it makes a lot of sense to inhibit both those arms and both those alterations, but we're hampered by having the right approach to target MIB at this point.
So as you can see, there's another question along those lines from Dr. Onikoko. The question is, great talk. What has been your perspective on immunotherapy for ACC and any impact of Notch or MIB alterations on that?
Yeah, I'm sorry, I had like, you know, 20 other slides to show you that I didn't have time.
By the way, I want to congratulate you for spending an hour talking on head and neck cancer and mentioning immunotherapy maybe just once.
Yeah, right. I had a whole section of immunotherapy because I know you can't get away with that, but so the bottom line with ACC, and I can even just show you the slide that's relevant, is this one for salivary cancers. And this is a figure from one of Luke Morris's recent studies on immunogenomics and salivary cancers. And so these three dots, this is salivary duct carcinoma, this is myoepithelial carcinoma, this is ACC, and this is everything else. This is melanoma here. This is lung squamous cell carcinoma. And what we're plotting out here are mutational burden, and by RNA-seq immune deconvolution, immune infiltration scores. And so the first thing is that there's heterogeneity among salivary cancers and subtypes, as you might imagine. Salivary ducts for instance, are quite hot, have a hot immune TME and a little bit of higher mutational burden. But a higher immune TME even compared to melanoma and lung squamous cell carcinoma. And ACCs here are both cold and have a low mutational burden and low PD-L1 positivity. But one of the interesting things that came out with ACC, and this is from Luke's Nature Medicine paper last year, is that the bottom line is, even in these low mutational burdens, you can find patients with T cells that are directed to the neoepitopes created by the gene fusion. So there's at least neoepitopes that we can target. What we need is better immunotherapy approaches. And indeed, the immune checkpoint studies that Roger Cohen published with pembrolizumab and salivary gland cancers, and the Nivo-IPI study that we had done here and reported at ASCO shows that the response rates with immune checkpoint alone are quite low. So the approach that we're testing in ACC and non-ACC salivary gland cancers is a lenvatinib pembrolizumab approach. That's the ongoing trial that we have here, hoping that the TKIs can improve the immune TME and improve immune checkpoint responses. Now, even though the responses are quite low, among the most dramatic ACC responses I've ever seen in a clinical trial, having done a number of these studies, has been when immune checkpoints work. So we're really kind of going for that, more impressive response with immunotherapies. We just need the better combinations to do it. Yeah.
So it's 8:31. We'll take just one question, one last question from Dr. Shin, which is, do you see any role of adjuvant therapy in saliva gland cancers?
Yeah, that is one of our most common consults is, would we add chemotherapy or systemic agents to post-op radiation? And the first is to kind of highlight that there is an RTOG NRG study looking at this question, taking high-risk salivary gland cancer malignancies that Christina Rodriguez and David Adelstein is running through NRG, and asking the question of comparing radiation versus radiation plus weekly cisplatin. So certainly, the results from that trial are going to provide some important insights. And in the end, without that data, it's really hard to provide guidance. When you look at large cancer registry kind of retrospective studies, actually, it's very hard to delineate a benefit to adding chemotherapy to post-op radiation. Off-label, we sometimes do it for patients who have really high-risk disease by different pathologic features or recurrent diseases that have been resected. In salivary duct, there's some data for androgen deprivation retrospectively demonstrating some benefit in that setting. And then lastly, I didn't get a chance to talk about HER2, but Glenn Hanna at Dana-Farber looked at their experience with HER2 targeting in the adjuvant setting, showing data that suggest a benefit. But what's really needed here are prospective studies, and those are ongoing and coming.
Well, I want to thank Alan again for a superb talk and a superb symposium. And I want to remind you that next week, our grand round speaker is Dr. Marina Garasino from the NCI of Milan, who will be presenting on COVID-19 and hyperprogression and lung cancer, clearly a topic of interest. And so also reminding you that to view all the Winship Grand Rounds lectures, please visit Grand Rounds page on the Winship Cancer Center website or the Winship calendar. Thank you very much, Alan, and we'll see you hopefully in person in a not too long time from now. Thank you so much.
Thank you so much for having me. Appreciate it.